My name is Rashpal Sagu, and we'll be going over the role of epigenetics has in the diagnosis and treatment of human diseases. Annie and I will be going over the basis of what epigenetics is, while Leanne and Lauren will go over how they are being used in the treatment of these diseases. Due to the extended debate on what the true meaning and functionality of epigenetics is, it's hard to pinpoint a true answer.
The basis of epigenetics is this. Although cells and organisms have the same genetic makeup with regards to DNA sequences, they still develop different phenotypes based upon the environmental factors.
Epigenetics is derived from the next generation sequencing, or NGS for short. NGS is the modification of genomes of many species. A popular method of implementing epigenetics is through DNA methylation. Through the DNA methylation process, a methyl group, rather than a carbon chain, replaces a hydrogen atom usually in the hydroxymethyl cytosine area, reprogramming the cell.
Prions are another example of epigenetics. Rather than targeting pyrimidines like DNA methylation, it arises through protein folding. These prions are able to survive in many different conformational stages. While they're also able to replicate themselves, they resist change to the environment, usually stress-related as needed. Just like many other proteins, extremes in temperature and pH can affect the rates at which prions operate. This usually leads to misfolding of the prion formation and changes the active site.
Epigenetic changes can be compared to switches because they are capable of turning on or off the expression of a gene. These changes occur due to addition or deletion of a methyl group or histones from DNA. If epigenetic changes occur, it is usually during gestation, neonatal development, puberty, or old age. These changes are commonly illuminated by humans with cancer. Tumor suppressor genes can be turned off by a genetic gene. Furthermore, overexpression of imprinted oncogenes, which are genes that have the potential to cause cancer, is frequent in cancer in humans. Cells contaminated with T-cell leukemia type 1 virus have shown abnormal methylation on the short arm of chromosome 11 in a region that harbors tumor suppression genes. In colorectal cancers, which are cancers that start in the large intestine or the rectum, have shown significant DNA methylation loss. In the same patient with the colorectal cancer, they have a normal amount of DNA methylation loss in the mucous membrane. Cancer is not the only case of epigenetic changes of imprinted genes. Epigenetic changes also occur in and have been most commonly researched in hepatitis B virus, human papillomavirus, and Epstein-Barr virus. Research has shown that it may be possible for individuals predisposed to disease due to epigenetic changes to prevent epigenetic changes with folic acid or genistein taken during gestation. Dr. Driddle uses computer learning algorithms that discover genes with high probabilities of becoming imprinted in humans. When they find which possible imprinted genes are actually imprinted, then they will be able to look at any condition and be able to tell which genes are deregulated and where. He said that eventually you could even make a chip and screen all of them. Treatments such as azacitidine and valproic acid work to fix the DNA methylation or histone modification. These treatments take away the epigenetic mark, which allows the growth regulatory genes to function properly. Looking for histone and methylation changes in DNA could be used as a diagnostic tool. However, at the moment, looking for genetic mutations is the common practice. Research is also being done on analyzing methylation on a large scale with diseases like bipolar disorder and autism. More knowledge could lead to new treatments since epigenetic changes are reversible compared to non-reversible sequence mutations in diseases. For example, DNA methylation/histone inhibitors have been used in myelodysplasia, a blood condition, in clinical trials. However, certain inhibitors, such as deoxycytidine, which is being tested to activate silenced tumor suppressor genes, can deactivate just as many genes as it activates.
The definition of epigenetics states that it is a non-stop, multi-directional exchange between both environment and heredity. Proteins are produced under many different environments within a lifetime. This theory, in most basic terms, is the argument of nature versus nurture. What are the determining factors that make a human body function the way that it does? More specifically, what affects the very basic outcome of life? Genes are being experimented with to see if their product can be altered without changing the basic code. Currently, research is being done based on the ideas of epigenetics in order to find treatments for a wide spectrum of diseases. For example, in the brain, neurons encode information and affect memories at the cellular level. Yet while memories may last a lifetime, neurons are dynamic structures. Every protein in the synapse undergoes some form of turnover, some with half-lives of only hours. How can a memory persist beyond the lifetimes of its constitutive molecular building blocks? Epigenetics changes in gene expression that do not alter the underlying DNA sequence may be the answer to treating memory degenerative diseases such as Alzheimer's. Another way that epigenetics can be integrated into the treatment of diseases is through its use to treat kidney-related problems. Regulated gene expression by transcription factor networks is critical for normal kidney function. Disruption of these complex networks leads to biochemical aberrations associated with many renal diseases. Epigenetic mechanisms not involved in changing DNA sequence, such as DNA methylation and post-translational modifications of nucleosomal histones, also play a critical role in gene regulation by modulating chromatin access to cellular machinery for transcription. These epigenetic modifications can be affected by intrinsic and extrinsic environmental factors and play a central role in dictating biologic phenotypes, including pathologic disease. Emerging evidence also suggests, apart from traditional genetic predisposition, that epigenetic processes can persist across generations to play a modulating role in the development of renal diseases such as diabetic nephropathy. Recent advances in epigenome research has increased our understanding of epigenetic mechanisms involving renal dysfunction that in turn may lead to the identification of novel new therapeutic targets.